So, hello everyone. It's my pleasure to share this session today on biology and pathogenicity. It's the second part of this huge session that was already take place yesterday. So, we will begin this session by a talk given by Ajaccio della Coleta Filho from the Centro de Citricultura Silvio Moreira in San Carlos in Brazil, and he will present his work on the spatial distribution and genetic structure of Xylella fastidiosa subspecies Poca in olive trees in southeast Brazil. The floor is yours.
Thank you. Thank you, Jax. Okay, I'm not Najla. Okay, I'm Ajaccio. Najla is my student, and is my master science student. The data that you're talking here is about part of her dissertation, and it's the preliminary analysis of the data that's collected in the sample. Okay, also there is some important collaborators in this work, like Professor João Lopes from Exalc, University of São Paulo, and also Luis Fernando from EPAMIG. EPAMIG is a research center in Minas Gerais state. Okay, so we are working together with this project. Okay, let's figure out this one. This work. Okay, this project is part of the task 2.2 from XF actors, and about population genetics of Xylella strains relevant to EU. Why it's relevant to EU? Because there you are working with, you have Pauca, but not the XT63, or the other ones ST from Pauca. Okay, so just talking about Xylella in Brazil, you have two different subspecies, the multiplex and the Pauca subspecies. The multiplex is important to it's causing disease, and it's a great treat for plumes productions. Okay, but the most important for us is the Pauca subspecies, because the Pauca is spread everywhere, the north to south of Brazil. East and west also. So it is important because it causes disease mainly in sweet orange. The sweet causing the seeds variegated chlorosis is the most more studied pathogen in Pauca in Brazil, but also cause problems in coffee plants. But the coffee plants, the economic importance of the Pauca in coffee, it is not well studied, for us not well quantified. Okay, and recently there was described in olive plants in Brazil. It's small industry growing in Brazil right now, but small, but is still causing problem in olive trees. Okay, and also you are described these subspecies in some ornamental plants like hibiscus. But it's a few specific cases. Okay, just talking about Xylella fastidiosa in olive. Okay, subspecies Pauca in olive. You can point here. Do you know? Oh, yeah, I think it's no. No pointer. Oh, okay. So, you can see this. Can you larger the map? Can you larger the map or not? No. Okay. So, no, because yesterday sometimes happen. Okay. So in the map. Okay. Okay, thank you. So in this map here, you can see from the areas that olive is growing in the northeast of São Paulo state. I mean São Paulo, here São Paulo state, Minas Gerais state, and Espírito Santo state. here you have Rio de Janeiro state, but you are sampling our samples from here. From here to here is almost 1,000 kilometers, okay? So the disease is spread for all this area here. We first reported in 2016. Okay, you can see here the yellow pins is the negative work shards for Xylella, and the red pins is the positive sites for Xylella. How you can say that's almost 85% of the work shards that you sampled has at least one plant with Xylella infection. So it's widespread, this disease. It's even you are recently reported. It means that it's stay there for more time that you thinking about this, okay? And another way to explain this one, in a short time you have a huge spread of the disease for a large area. Also, there is another things that we can talk about. We not talk about this one. Okay. In the beginning, we started to do sampling with this analysis to see how not how the spread the disease, but what is the MLST loci was present in those populations, and you will figure out that you have the conclusion that the XT16 was present in almost 75% of the samples that you analyzed has this ST16 over here that was first reported in coffee plants. But you can find another ones like this STs, 8 new ones, 85, 86, and 84, 5, and 6 that was reported in one paper on phytopathology pathology recently. But when you go forward with the sampling, you can find more STs, like this one here, ST13, that was first described in citrus plants, and also the ST70 that was described in hibiscus plants. On a part of our short history, we take some different STs from that you found in the field and also that you didn't find, but it was present you are reported in citrus and coffee, and in fact, olive plants in greenhouse. Okay, as you can see here, most of the STs from Pauca, even they came from citrus, even they came from coffee, or specifically from hibiscus, they are able to infect olive plants in greenhouse by artificial infection. In different ratio, but you can affect this one. And it's, of course, that you must go by higher this number over here, but it's interesting that this ST71, this even it's Pauca, but was isolated from Planus, and and this one that was isolated from Planus also was not able to infect olive plants by artificial infections, but just strongly, but you must go doing for more infections over here, because you have a few samples, a few events of infections. Okay, it's not means that it's so early to say that the isolates that came from plants is not able to infect olive. It's so early to say that one, but just stress about these results here. Okay, back in our history, here it is the olive orchards that you analyzed from Minas Gerais and São Paulo state. They are present in different altitudes. They are they are present in different altitudes and the variable landscape vegetation around the orchards. So you have no questions about these populations. How could these different environments could affect, or there is effect or not, about the genetic diversity of the bacterium present in those different altitudes and those different landscape vegetation? Okay, that was nice question. What is the distribution of the genetic diversity of the Xylella populations across an hierarchical of geographic scale in those populations? You are talking about different altitudes, you are talking about a huge area that was infected by Xylella in olive plants. To do it, you must use molecular markers. Of course, if you are able, you could do a deep sequencing, but it's not a deep sequence, but a whole sequence of the bacteria, but you are not able to do it in population scale. So you're using the more fast evolution markers that you have, this VNTR or short sequence repeats. Okay, it's more adequate tools to have to analyze in the population at the strain level. So to do it, when just work for Xylella fastidiosa isolation, you go to the symptomatic plants, choose the asymptomatic branch from the symptomatic plant, okay, closer to the symptomatic branch, cut this branch over here, doing disinfection by bleach and alcohol until you plot in medium. Okay. After you have the colonies, we select just one colony, okay, and start to grow the bacterium and keep in the cryogenic collection, and part of this goes to typing of the bacteria. So all those isolates that you're working on, you have in our collection, cryogenic collection in main 18, minus 18. Okay. So, and for the microsatellites, you working with 4 different sets, and each set you have 3 different microsatellites, and the microsatellites or the 4 departments are marked with different fluorophilus, and we're able to do in multiplex, okay, even by PCR and even by fragment analysis. The analysis that you are doing, not finished the statistical analysis, or you are, the data that you are shown here, it's about the genetic, general genetic informations, the number of different alleles by loci, by locus, different patterns, allelic patterns, number of genotypes by population, genetic diversity. So you're doing a principal coordinate analysis just for figure out about how the strange distributions in the populations. You are using here genetic distance among all the individuals or among all the strains, and also you using try to figure out about the cluster, genetic clusters inferred by Bayesian statistics, okay, using structure. And so this number here is not 1 to 15, but 1 to 20 that you run the structure. Okay, about the results you have here, you can see about the number of different genetic haplotypes you have in the front, the 177 strains that you have, that is in our collections. You have 72 different genotypes, microsatellite genotypes. It's assuming that each different markers is one genotype. You have one specific genotype. You have 72 different genotypes in the population. And the genetic diversity is quite variable among the different populations. You have 17 different populations, 17 different places that you collect the samples. You can see here. For example, the area, you name it MG A1, okay? You have 35 different strains that comes from there, okay? The different number of the alleles, but I stress about this genetic diversity. It's quite variable among the populations. Some populations are clonal, It's Vivian, you have few individuals, but it's clonal, but you have here, for example, a significant number of the individuals, but low genetic diversity, okay? But some populations, the genetic diversity is high, some ones it's low. In the overall, the genetic diversity of the population, it's low. If you compare with populations from CVC, from sweet orange, the genetic diversity is high, and from the coffee leaf scorch, the population is high also. It means that it means not, but this population is already established for a long time in the field. This one, it's a recent outbreak. Okay. About the principal component analysis, you can see here 3 different clusters from the 17 different populations. You can see 3 different genetic clusters. One main cluster, cluster 1, is represented by most of the strains over here. Okay, almost 90% of the samples were grouped in this cluster over here. Okay, you have the second one that is composed by 4 different geographic populations, individuals from 4 different geographic populations in mixture with samples from the cluster 1. And also in third cluster, here this is composed by one well-structured geographic population. This came from all the population from here, came from one specific place. Okay, over here. It's important to, not important, but it's interesting that to mention that the most of the strains from the cluster 1 comes from the area A1 and A2. That is, I can say, one main irradiation nursery plants from those growers. Okay, we can figure out about what's happened here. So by the structure analysis, you can see also similar results that can see by principal component analysis. You have 3 main clusters where all this cluster was have a good correlation between the component principal analysis results, except the cluster 3 here from the structure analysis that we have one population from area MGA5, but in the component analysis those not present there. Only the individuals from this area here are presented in the main cluster 1 in the component principal analysis. And also by AMOVA analysis of the molecular variance, you can figure out that the main genetic difference is from among populations, and you have one strong, okay, and with strong index for difference between the 3 different populations. Okay, in conclusion, the low genetic diversity index was observed in this population that could be explained by the recent outbreak of Xylella causing olive quick decline. Okay, and then another one, oops. Okay. Oh, come on. So far, the local characteristics of the natural reserve of the specific environment have no influence on genetic structure of the bacterium, with few exceptions. Populations from 17 geographic areas, regions were clustered in 3 genetic clusters. And you can, but clonal dissemination of bacteria by infected vegetation material could not be discarded. Okay, that's one of the main conclusions about this one. So we are very thanks for the XF Actors project, and also for São Paulo Ajaccio by the grants. And I would like special thanks for the olive farmers that have possibility to sample the material in their properties. And also important people are behind this data, my student in Ajaccio, and my ex-technician Kelly that doing all the works, the bench works. Okay, thank you for your attention.
Thank you, Helvecio. No, there is time for one question. Is there any question for Helvecio? One question, yeah. Is there any question for Helvecio? So I have one. I saw in your isolation workflow that you isolated strains from asymptomatic material. I wondered why you did that, if that you do not have access to a lot of symptomatic material.
Yes.
And how does it impact, finally, the diversity of the strains you isolated?
I mean that I take the symptomatic branch closer or behind or below the symptomatic, okay? Because it's more easy to isolate the bacteria.
Okay, but do you have an idea of the impact it can have on the genetic diversity of Xylella you isolated? Is there any evidence that the diversity of the strain is exactly the same in the asymptomatic part of the plant that in the symptomatic one?
No, I have no idea, but I suppose that that no influence, because just having I use this one here. Imagine that you have the disease over here, okay? The symptoms, you take the sample over here, okay? Because most of the branch here, it's almost died. So they take the sample from the live branch. Okay. I think it's, I have
okay,
no
yes.
sure, but
Thank you very much. Thanks. So now, I'll call Lindsay Burbank from the Agricultural Research Service, USDA. She is in Parlier, California, and she will present her work on the cultivar susceptibility and temperature-dependent recovery of Xylella fastidiosa infected grapevine. So.
Okay. So first, I just want to point out that the experiments I'm talking about are controlled greenhouse experiments. This is not field data. So I'm doing this for the purpose of looking at some very specific ways that temperature can affect disease progression and then vine recovery the following season. So all the experiments that I'm going to talk about are done in a controlled environment. And these are focusing on grapevine and subspecies fastidiosa. So everything I'm talking about is subspecies fastidiosa. Okay. So I'm doing this work in California at the San Joaquin Valley Agricultural Sciences Center. And where we're located is right in the middle of California. So we're right in the middle of the valley. It's a large agricultural area. And we have different climate in the valley than we do on the coast and in some other regions of California. So a lot of what I'm talking about is going to be focused on this area. But again, the information is from experiments in a controlled environment, so it can easily be applied to other places with a similar climate. So I'm going to talk about vine recovery. So this is when grapevines are infected with the pathogen, and then the following season, they are no longer infected with the pathogen. So in many cases, they can actually recover over the winter. And this has to do with the winter temperatures in the region. It also has to do with the cultivar, and it also has to do with the summer temperatures and how advanced the infections are before they experience the cold. So we've known about this kind of thing for a long time. Grapevines can recover from the infection over the winter. And this goes all the way back in California about 20 years. This is work from Purcell's group at UC Berkeley. And what they found here is that time of inoculation is very important for the likelihood of vine recovery over the winter. So if you have vines that become infected late in the season, they're much more likely to recover. If you have vines that are infected early in the season, this often leads to chronic infections, and they will come back the next year. Now, a lot of these experiments were done out in the field in the northern areas of California. So up on the north coast, the Napa-Sonoma regions where they grow a lot of wine grapes, and then as far south as Fresno County, this is where Parlier is located. So some of the experiments were actually done outside in Parlier. Now, this is not the only place in California where we have a lot of grape cultivation. So we grow a lot of table grapes in the San Joaquin Valley, and especially in the southern areas of the San Joaquin Valley, Pierce's disease has been a big problem. And then as well, down even farther south, the Temecula Valley, they grow wine grapes there, and they had large problems with Pierce's disease. Now, we have different climates across the state of California. This is just one measurement of winter temperature. So this is the accumulation of what we call chill hours, which is just the number of hours below 45 degrees Fahrenheit, which is roughly 7 degrees Celsius. So it's not an extremely cold environment, but it's enough for the grapevines to go dormant, and it's enough to sometimes control Xylella. Now, I do have to mention that if we were looking at this outside in California, we do have different vectors in Northern California and in Southern California. So the behavior of the vector and how this influences the disease dynamics would be very important if you are looking at this out in the field. So speaking about in the field, these are some pictures from 2016, 2017 of areas that were affected by Pierce's disease. So you can see early in the season, you can see the places where the vines were removed the previous season because of disease. But when they do this in California, often they test for the disease during the summer, they figure out what's infected, and then they come back several months later during the winter when the vines are dormant, and that's when they remove the vines. So you can see they're removing the vines during the winter, there's no leaves on them. So vines that are known to be infected often are out in the field for a few months before they're removed. Now, we also know that it's easiest to detect the pathogen during the summertime. So I was part of a project in this area with some of my entomology colleagues who are looking at what is the time of year in this southern part of the San Joaquin Valley where you get the highest percentage of insect vectors that are carrying the pathogen in the vineyards that are infected. So we looked at the plants. So we tracked infected plants over the course of the season. We also monitored the insects, and we would collect the insects and test them for the pathogen. And it's late past the end of July and into October, this period of time, is when you get the highest percentage of insects carrying the pathogen, and this is when you're most likely to be able to detect it in the plant. So this period of time is important if you're looking at disease in this area. This is also when you see the symptoms. So this is when you can go out and you can look at the scorching and you can identify Pierce's disease out in the field. Now, this period of time is also, if we look at these earlier studies of overwinter curing and vine recovery, this is the period of time where almost 100% of vines in some regions, if they became infected during this period of time, would recover. So it's likely that in the southern areas, this is not really what's happening. There has been some more recent work that actually does show that you don't have a significant effect of this vine recovery in the southern parts of California. But I was interested in looking at what the specific temperature effects are during that time that might affect the disease and then the vine recovery. So winter temperature does have an impact on this. In California, we don't get extremely cold winters, so I'm mostly looking at duration of a lower temperature here. And so these are some inoculation experiments done in the greenhouse where the plants are infected, they're tested, they're positive for Xylella. Then I put them in a cold room at about 4 degrees for certain periods of time and look at how many of them recover after they come out of the cold room. So if you can correlate the number of chilling hours or the number of hours in the cold room with the regions, if it's about the amount of cold that would be experienced in the Temecula region far south, you really don't get any recovery. So the vines come out, you grow them back, and they're all still infected if they were infected previously. If you leave them a little bit longer, so this is the southern portion of the San Joaquin Valley or Kern County, you get some recovery, but it's still at not very high rates. But if you leave them in for even longer, equivalent to what would be experienced in Northern California, you can actually get pretty high rates of recovery. in potted grapevines that have been inoculated. So this is similar to what was previously seen in the northern areas anyway. But this is all done on a susceptible cultivar. This is Chardonnay. We like to use it for experiments because it's highly susceptible. But they can also influence how well the vines recover, depending on the cultivar. So we also wanted to look at a wider range of cultivars, things that we hadn't necessarily looked at before. And in California, we have a lot of wine grape production. It's a large portion of the grapes we produce. But we do also have table grapes and raisin grapes. And if we're looking at specifically the San Joaquin Valley, we have a lot of table grape production there. So this is something that can't really be ignored if you want to look at how well the vines out in the field might recover in this particular area. So I included more table grape varieties in this study. This is only part of the data that I have. If anyone is interested in this, I'm happy to answer questions about that later. But I pulled out these two varieties because you can see a clear difference. So Cabernet is something that I chose because it was the subject of a lot of previous work on Xylella in California. So we have a lot of information about it. And if you look at how well Cabernet recovers after about 4 weeks in a cold chamber, you get pretty high rates of recovery. So that's very different than some of the other cultivars I looked at. The other one here is Scarlet Royal. This is a table grape variety. This was actually developed in Parlier in California. And it was very popular for a number of reasons, but it was also very susceptible to Xylella. So what we found with Scarlet Royal in particular is that pretty much all of the vines that were infected, they remain infected. It doesn't really seem to have this recovery effect the way that some other cultivars do. But I also wanted to look at something that we don't really have a lot of information about as much as we do about the cold temperature, which is how the temperature during the summer can impact this recovery effect as well. Because if you go to the coast of California and then you go to the Central Valley of California, these are very different in the summer. It's very hot in the Central Valley, and this can affect how the disease progresses and potentially how well the vines might recover after that. So if you look at the daytime temperature during the summer, in California it's very hot, and you can see if you go up through September and October, in the southern areas it stays that way. So it stays well above the temperatures that would be good for replication of Xylella and that would be good for Pierce's disease. So you could potentially have a much longer period of time where the bacterium is active and you're getting an increase in infection in the plant. So if we go back to this original idea that time of inoculation during the season is important for vine recovery, we have to separate what is the effect of the duration of infection and what is the effect of severity of infection, which could be potentially influenced by the temperatures during the summer. And we know this because over and over again in greenhouse trials, and if we go out in the field, the symptoms show up when it gets hot. We see this effect, the higher the temperature, the faster this can progress when we're looking at greenhouse trials especially. So we know that heat has an influence on disease in grapevine at least. So I set up some experiments to separate the effect of time and the effect of temperature. So these are several different sets of plants that were inoculated with Xylella, susceptible variety. And I separated it into 2 treatments that experience an 8-week inoculation period and then one that experiences a 16-week inoculation period. And the 2 8-week treatments are different in that they're inoculated at different times during the season. So one is going to be in June, and then the other is going to be inoculated at the end of August. So the infection period would be during September and October. Now, if you look at the 2 8-week treatments, they experience different temperature conditions. So they're in a greenhouse, but the temperature in the greenhouse is still impacted by the temperature outside. It's only minimally controlled temperature-wise in there. So the first group, you get a very rapid increase in degree day accumulation, and I use 25 degrees as a baseline for replication of Xylella. But you get a significant increase above this temperature during the day if it's during the summer. So these are the plants. The first group is plants inoculated in June. Then the second group is inoculated at the end of August. It's starting to cool down outside, and you don't get the same increase in temperatures. It experiences a much lower average temperature overall during this period, so you don't necessarily get disease progression as fast. And I have data on the symptoms as well that I'm not going to talk about here, but it does affect the extent of symptom development during the experiments. So after the end of the inoculation period, all of these vines go into a cold chamber. They experience the same period of cold treatment, which is 4 weeks at 4 degrees Celsius. And then they come out and I grow them back. And this isn't long enough period of time for the vines to go dormant. They have no leaves on them. And so when I'm looking for reinfection, it's the new growth that comes back from the grapevines. So again, there's an effect of cultivar. So Scarlet Royal is a cultivar that doesn't seem to recover very well at all. It doesn't really matter what treatment it experiences. 8 weeks is enough time for it to become infected enough that it's not going to recover. But you do see some differences if you look at other cultivars, such as Cabernet, where 8 weeks early in the season, where it's much higher temperature, is different than 8 weeks later in the season, where it's a much lower temperature. And so these are the same period of time, but they're a difference in temperature. And then a 16-week treatment, most of the vines remain infected. So it does matter how high the temperature is during the summer when you're going to look at the effects of recovery out in the field as well, very likely. So vines can recover out in the field sometimes due to temperature, but this is dependent on both the winter temperature and the summer temperature, and is also going to be very dependent on cultivar. So you have to consider all of these factors if you're going to look at a field scenario, because it can be very different depending on what you're looking at. So that's all I'm going to talk about for today. If anyone is interested in the rest of this data that I have more varieties and more treatments and a lot more information that I didn't have time to present. I'm happy to answer any questions about that. But I would like to thank the members of my lab in Parlier who helped with this work, and also my collaborators in our research unit who have been involved with a lot of Pierce's disease research and vector research. And this is funded by the United States Department of Agriculture as our base funding. Thank you.
Thank you, Lindsay, for this very interesting talk. Is there, yes?
My question is, are there any differences in population levels between recovered and non-recovered vines?
There don't seem to be very big differences in population level. You can detect the pathogen in the plant. They're at a high enough level to detect. There is differences in symptom development. So the plants that have more advanced symptoms are usually less likely to recover.
Another question? Yeah.
In terms of the experimental chills, is it a continuous cold or is it more cyclical? Because I imagine that could have a pretty big difference on the ability to grow if you're getting warm every day.
Yes, so that's a good question. This is a continuous cold treatment. It was done that way for the purpose of simplification. So it's based on the chill hour accumulation data that we can collect from out in the field very easily in California. But yeah, there are likely going to be differences if it's outside and you're experiencing significant increases in temperature during the day versus at night. So this is a simplified version of this. Of course, it would be different out in the field, but we might still be seeing a lot of these effects out there as well.
Do you have any clue about what are the reasons of the difference in recovery between cultivars? Are they related to silent anatomy or anything else?
I don't really know the answer to that. A lot of the cultivars I included in the studies were part of our breeding program. We have a breeding program for table grapes. So that's a question for our table grape breeders, I think. We don't necessarily have a lot of information about many of them.
Just a curiosity about recovery. When you speak about plants that recover, you mean that in the new growth you don't have isolation of the bacterium and symptoms? And what about the old part of the plants? You still is detectable, or it became undetectable?
So when the plants are in the cold, they go dormant, the leaves fall off, they're pruned back to a single cane. So I haven't actually tested the trunk, the stems. So all of the testing I'm doing looking at recovery is from the new growth that comes back the next season. But I leave them growing for about 6 months. And so most of these cultivars are susceptible to some degree. And so if you do have an infection, you would usually see it over the course of 6 months.
To follow up on that question, after 6 months, you can detect the bacterium by qPCR, or you just observe symptoms and note symptoms?
So this is based on symptoms, qPCR, and also culturing of the bacteria.
Is there another question? No? So, thank you very much. So now we have the pleasure to have Miguel Roman Escija from the Institute for Sustainable Agriculture in Cordoba that will present his talk on the phenotypic characterization of 2 Spanish strains of Xylella fastidiosa subspecies multiplex, ST6, that are differing in plasmid content.
Thank you very much. First of all, I would like to thank to the organizing committee of the Congress for select my communication as oral presentation. This research is part of my PhD thesis that I currently am doing in the Institute for Sustainable Agriculture in Cordoba. And Xylella fastidiosa was detected for the first time in Milan, Spain, in July of 2017 on the East Coast. Since then, several outbreaks of the bacteria has been detected in the demarcated area that currently covers more than 130,000 of hectares. In the infected plants, so far, only subspecies multiplex sequence type 6 has been detected. In previous research, two strains isolated from almond tree were isolated from 2 different zoocards, and they were designed as EVIA 59-1 and ESBL. The genome analysis of both strains reveals that they share more than 99% of the chromosomal genome, but however, they differ in the presence of 2 plasmids in the ESBL strain. One big plasmid with a high similarity with a plasmid from subspecies Pauca, and another plasmid smaller with a high similarity with a plasmid from subspecies fastidiosa. Further research in our lab group are concerning about the incidence and the diversity of the content of plasmids in a set of samples on almond tree in this area. For more detailed information, have a look to the poster that my lab colleague Pilar has presented. Colonization, and this is development of the bacteria in host plants, depend of several factors: cellular adhesion, cell motility, biofilm formation, and the size of the aggregates. These factors are usually mediated by filamentous structures called pili. Xylella has 2 types of pili: the pili type 1 which is shorter and usually related with biofilm formation and cell aggregation, and the pili type 4, which is longer and usually related with motility, with cell motility. There are also another structure called non-fimbrial adhesins that also could play an important role in all of these processes of colonization and disease development. In this context, the main aim of this research is to characterize phenotypic traits associated to infection and disease development of these two Spanish strains of subspecies multiplex sequence site type 6 to find out if their difference in plasmid content could be related to the difference in phenotypic traits. For this, to assess phenotypic characterization, we have performed several experiments: patterns of bacterial growth, cell adhesions experiment, biofilm formation, cell-cell aggregation experiment, twitching motility, and a pathogenicity experiment using 2 tobacco cultivars. Spanish strains were compared with 2 strains from subspecies multiplex, ALMA and BB08, and with the reference strain of Xylella fastidiosa, subspecies fastidiosa, Temecula 1. To assess patterns of bacterial growth and cell adhesion experiments, we use microfluidic chambers as a model simulating silent vessels. This microfluidic chamber has 2 parallel channels, and that allows us to compare 2 treatments, or in our case, 2 strains. And with an special software, we can take photos periodically in a time lapse of 30 seconds. And that allows us to have a continuous tracking of cell behavior over the time. With this system, we can observe the cell's movement and how cells form aggregates and biofilm. Our results using microfluidic chambers shows that the strain 59-1 seems to have almost no mobility. Only some cells can move poorly. In contrast, the strain ESBL and Temecula 1 cells can move. As you can see in the video, you can see some of the cells with some difficulty, some of the cells going against the flow. And we also have calculated the speed of the cells, and we have seen that Temecula cells have a higher speed. And we also have observed that the Spanish strain form the same type of aggregates. Both strains form small aggregates that can occupy all the channel over time. And with the same design of microfluidic chambers, we have assessed the cell adhesions of each strain to a glass surface. This experiment began with a low flow, and by a continuous increase of this flow of 10 microliters per minute, we can determine the number of cells that are attached to the glass surface. And with this data, we can calculate the adhesion force. As you can see in this picture, around 100 microliters per minute, most of the cells of the strain 59-1 were dragged downstream, while most of the cells of the ESBL strain are still attached to the glass surface. As we can see in this graph, the adhesion force to the strain ESBL is higher, and that means that this strain has a high capacity of attachment to the surface glass than the strain 59-1. We also have tested biofilm formation with the crystal violet protocol, and we did it with, in order to consider the variability of this kind of experiment, we did 3 independent replicates for 3 repetitions. And our results shows that Spanish strain has less capacity to form biofilm compared with the reference strains, and there are no difference between Spanish strain in biofilm formation. We also have calculated the settling rate, which is the ability to form cell aggregates. When cell aggregated, sedimentation is higher, and aggregates settle at the bottom of the tube. We estimate this rate measuring the optical density at 600 in the time 0 and an hour later. And our result shows that Spanish strain, again, has less capacity to aggregate cells, and there are no difference between both strains. We also have tested the biofilm formation in crystal tubes, and we have seen that all strains form the typical ring, and also we have observed that Spanish strain form more turbid suspension, cell suspension, confirming the low capacity to aggregate cells. This low capacity to aggregate cells maybe is correlated with the low capacity to form biofilm in the microplate experiment. Another trait we have measured is the twitching motility, which is the extension, the study of the extension of the retraction of the pili type 4. We have observed the morphology of the edge of the colonies at 2 and 5 days, and colonies with a peripheral fringe, marked with a yellow arrow, were designed as twitching positive. Our result shows that the reference strain, ALMA BB08 and Temecula 1, has fringe at 2 days, and it is higher at 5 days. And regarding Spanish strain, we have seen that the strain 59 and 1 has no fringe at any time, and also has a smooth margin in the colony. While the strain ESBL only has fringe at 2 days, and also we have observed a singular morphology of the edge of the colony, which is a granulated margin. Finally, I'm going to explain the pathogenicity experiment. We have inoculated 4 isolates in 2 tobacco cultivars, Petit Habana, SR1, and Shanti. For inoculation, we put 10 microliters of cell suspension in 2 petioles per plant, and we prick it with an entomological needle. We made this inoculation twice. To assess disease severity, we count the number of leaves with yellow wing and scorch symptoms weekly after the first symptoms appears. And our result shows that all isolates were pathogenic in both tobacco cultivars, with the exception of the strain 59 and 1 on the cultivar Petit Habana SR1. And regarding Spanish strain, has a lower disease severity compared with the reference strain Temecula and ALMA. With these results, we obtained the following conclusions. Spanish isolates has less motility, ability to form biofilm and cell aggregates, and virulence on tobacco compared with the other reference strains. Compared with the strain 59-1, the strain ESBL has higher motility and adhesion force, and it is pathogenic in both tobacco cultivars. Our results may indicate that Spanish strain don't have PILI4 or lack of some of the functions related with the formation of functionality of the PILI type 4. And this phenotypic difference might be caused by the presence of the 2 plasmids in the ESBL strain, as both strains are identical at more than 99% in their genome. Further research is needed to confirm if this phenotypic difference is due to the presence to the both plasmid in the ESBL strain. Finally, this research was developed in the laboratory of Professor de la Fuente in our university in the framework of the exec factors and with the support of Eurochamp cost. Finally, I would like to thank to all the lab group, to Professor de la Fuente, and of course to my lab group in Córdoba for helping me in all of this work. And thank you very much.
Thank you very much. Is there any question for Miguel? Yes, please.
Thank you, Miguel, for this very nice talk, and looking very in depth to the bacteria. I'm wondering if you repeat your experiment with other hosts, do you think the pathogenicity could be different? And also, from all these characteristics you were studying in the bacteria and these strains?
We don't know yet. We are testing this both strain in olive tree, in plants, and also in almond, and in fact in the VR center there are...
Use the microphone, please.
Sorry. We are testing this strain in olive tree and in almond tree too, but we don't have results yet.
Yeah, but what's your feeling? I mean,
I don't know. In almond tree for sure, because these strains are pathogenic in almond tree in Alicante.
Yeah, that's
but
why.
I don't know if they are
because
pathogenic.
seems to me it's a quite lazy bacteria in general.
It seems,
so.
but they are pathogenic and create disease in occurs in Alicante. So.
Okay, so we'll wait for your results on that. Thank you.
Is there another question? Yes.
Sorry, Miguel. Yeah, really nice talk. I agree with Professor Miranda. I have a question about, you say that there is no PILI4 in one of the isolates. Your results suggest that. Your results suggest that. I would like to know if you, because I think that if I'm not wrong, you have the genome completely sequenced that you published in January. Did you look if there is the genes that are able to form in the...
We are doing a preliminary study of the genome of the plasmids, and we found some genes that could be related with the formation of pili, and that maybe give to the strain factor of virulence, but we have to study more deeply.
Thank you. Another question? Thank you. Then. So now, Alexandra Kahn from the University of California in Berkeley will present her work on the ancestral state reconstruction of Xylella fastidiosa host-plant relationships.
Hi. Okay. Forward, back. Yeah? Yes. Okay, great. Got it. Okay. Thank you so much. Hi. Thank you so much for having me here today. It's really a pleasure. So I am going to talk today about my work using ancestral state reconstructions to try to analyze some aspects of host specificity and host breadth in Xylella fastidiosa. So, as we are all well aware, there's an extremely varied set of potential hosts, host plants for Xylella. And but with that being said, it's by no means a generalist within both subspecies, sequence types, and within individual strains. There's often a huge amount of specificity. So with that being said, the questions that I'm really hoping to address with my general research, I am as well a PhD student, so it's lots of work in progress. But what I really want to address is host specificity genetically determined, and with this particular project, can we use phylogenetics to understand aspects of it? So to start out, I'm going to do a little bit of phylogenetics review of some main important points from my talk. So
one of the main elements that I'm thinking about is congruence between trees. So you can see here 2 trees that I've drawn, one of 3 common hosts of Xylella and one of 3 sequence types within subspecies multiplex. So when we want to compare trees of the same data set, we want to know if they're congruent or if they're the same overall. And the 2 main metrics for that first are to compare branch lengths between the 2 trees, which in this case, the overall topology is the same, but the almond in pink has now moved closer to the plum, and representing fewer overall differences between those 2 taxa. And the other aspect is changes in topology, which are actual movements of the location of taxa compared to other taxa that we're interested in. And cumulatively, those are the interests that we have in overall congruence. So we're going to now get back to our original tree that I've drawn. And as you can see, the artificial branch lengths I've drawn, which are always apparent based on the taxa aligning at the tips. So the branch lengths between these are totally made up, but they do represent actual topologies. So when I'm thinking about aspects of the relationship between a host and a pathogen, I really think it's important to consider both sides of the question. So as has been discussed today and over the past 2 days, there's huge variation within plants, between cultivars, within a genus in terms of susceptibility. And so there's this aspect that we can see here, plum and almond are both within the same genus Prunus, and we want to know, because of the plant proximity to each other phylogenetically, is that going to lead to more similar overall susceptibility patterns to different pathogens? And that question doesn't depend at all on the genetics of the pathogen. It's just, are there phylogenetic patterns within the host relationships that are going to influence how they are susceptible? And then we have the other side. We have sequence type 6 and 36, which are sister to each other, more closely related to each other than either of them is to 24. And so on that side, we want to know, do they have more similar infectivity because they're closer related than either of them do to 24, which is more distant from both of them? So while these questions are intrinsically linked, when we want to know if something's going to infect something else, we have to consider both, and they really are distinctive questions. So just to demonstrate how these relationships can play out, I've laid out from some experimental inoculation data what these relationships might look like. And just to start out with our hosts, here, use this pointer. So we have plum and almond in Prunus genus. And we want to know, because they're more similar, do they have more similar patterns of susceptibility? And in this case, we don't see that. We see that plum is susceptible to these 2 and almond to these 2. There aren't any more similar in susceptibility to each other than either of them is to grape, which is only infected by ST6. And then we can go to the bacterial side of things as well. And we can ask, are ST6 and ST36 more similar in their capacity to infect, regardless of the plant phylogeny, in a particular host than ST24 will? And for this example, we see the same kind of patterns, which show that the phylogenetic proximity of ST6 and 36 doesn't determine their ability to infect. So moving on to how my project actually worked. So I was using a data set of 349 whole genomes with host information to at least the genus level, and as well as location data and, when possible, information about symptoms, but that wasn't as ubiquitous as the host information. So using that data set, we made 4 phylogenies of unique genetic loci. The first was based on a concatenation of the 7 MLST genes, which have been discussed a lot. Everyone's familiar with them, sure. But that's a concatenation of all of the nucleotides comprised of those 7 genes. The next one is our core genome tree, which is about 1,000 genes, and it's also a concatenation of nucleotides. And that adds up to over 500,000 base pairs. And that's typically what's considered the best genomic tree. It's the most thorough shared set of data between all of the strains of interest.
The next one is a tree that we actually removed the recombinant regions from that core genome and made an alignment of the regions that theoretically don't contain recombination. And the final one is kind of intrinsically different from the other three trees. Instead of a concatenation of a nucleotide alignment, it's actually, the character matrix is totally different. So the accessory genome, which is a representation of overall gene presence absence between the strains, the matrix is actually a list of all of the genes in any strain of Xylella, and then each character state will be whether a particular strain does or doesn't have that particular gene. So we have these 4 trees, and now we're really interested in understanding if they're congruent or not, and what they can potentially represent differently about the overall biology and pathogenicity of Xylella, like these different aspects of the genome. So this is an example of what our core genome tree looks like, and what you can see mapped onto it in slightly blurry colors and letters is the actual topological congruence between the core genome tree and the accessory genome tree. So the nodes that are closer to dark blue show that there's more congruence between these 2 trees, and when the colors fade closer to yellow, but I don't think any of them are any lighter than a light blue, it shows that these particular nodes are not congruent between the 2 trees. So what we can see actually overall is the accessory tree that we've made, which is this totally different data set from the core genome nucleotide alignment, has really, really similar topology to that original core tree. But the main difference, which isn't represented here, is actually a difference in branch lengths. So there's about an order of magnitude longer branch lengths on average within the accessory tree than there is within the core tree. So the method that I'm using for this project is ancestral state reconstruction, which I don't think a lot of people in this field are typically familiar with, so I wanted to introduce it a little bit. But pretty much the idea is that we have our genetic data set, which in this case is a phylogeny, and then we have our modern distribution of traits. And using that modern distribution of traits, we look backwards and try to understand what the most likely patterns of ancestral character states would have looked like. So this is an example having nothing to do with Xylella. It compares oviparous versus viviparous birth in squamates, so snakes and lizards. And using the tips, which they have the modern distribution of oviparous versus viviparous birth, they were able to look backwards and predict that oviparous birth was the most likely state for the ancestor of this whole group. So I did the same thing with Xylella, but instead of using birth type, I used host. So we have the same kind of data. We have a tree, and then we have the modern distribution of traits, or in this case, host, and we want to focus on these ancestral nodes and try to predict what the most likely pattern of host would have been at those nodes. So, one more thing I wanted to say, but I will say it now. To go back to our original questions of both the relatedness of hosts and also pathogens, I wanted to address that by using 2 different scales of plant hosts for the ancestral state reconstructions. So I did it both at the genus level, where each node and character that I use is a genus of the host, and also at a larger scale, these 2 superorders of astrids and rosids, which encompass almost all of the hosts of Xylella that we have experimental data on. So why these 2 groups? There are several reasons. The first is that they don't tend to overlap in susceptibility. And I don't mean that on a subspecies level or a sequence type level, because they absolutely do. But I mean in actual experimentation that people have done with cross-inoculation, there tends to be these distinctions, both in nature and in lab, between infectivity of plants that are in the astrid group versus the rosid group. So this first example is one that's been experimentally tested a lot and well understood, that there are these 2 separate strains that are able to infect coffee and citrus, and they don't co-infect. And other examples are, there have been a lot of experimental studies comparing Nerium oleander with both the genera Prunus and Vitis, and we see similar patterns.
that the same strain, one individual strain, is unlikely to infect both of these groups. We've also heard in the last couple of days other examples of these similar patterns in the field, often with olive, which is another asterid, not having, olive is frequently infected, and then in field situations it'll be coexisting with almond, which we know is susceptible to some strains, but is often not susceptible to those same olive strains. It's not a perfect delineation, but it's one that I find interesting. So for some actual results about this ancestral state reconstruction. You can't really see the colors on this screen, but this is our core genome tree, and all of the nodes have the most likely ancestor mapped onto them. So it doesn't really matter because you can't see it, because in this particular tree, all of the nodes that are beyond like the ancestor of only a few strains pretty much have a 50-50% likelihood of either of our groups. So this is representing, in the colors you can't see, red is the asterid host predicted, and blue is the rosid. And in this tree, the top branch, use the actual thing, this is pauca. In the middle is multiplex, and from this node on, this is all fastidiosa. So the ancestral nodes of each of these strains don't have a pattern that is shown to correlate with the phylogeny between host specificity. But then we also have this accessory tree, which we already talked about is the same topology as the core genome tree, but different branch lengths. And you can see it looks much more spread out, even just to the eye. But when you look at those important nodes, so I've highlighted the ancestral node of each subspecies, we actually do see that each one of them has a higher likelihood of being the ancestral state being an asterid host as opposed to a rosid host, so all three of them. The ancestor of all Xylella has the same 50-50 that the core tree does. But there are actually patterns that follow between the accessory tree and predictions of ancestral host state. So back to our original question is, can we use these different loci to try to understand relationships between plant hosts and Xylella fastidiosa strains? And in general, with most of our trees, so the core genome tree as well as the MLST tree and the core genome without recombination all look exactly the same. And for those trees, there doesn't seem to be any correlative relationship between the ancestral host range and the phylogeny, or our current genetic data. However, for this accessory tree, we actually do see patterns that track throughout history. So I think it's really important to acknowledge that Xylella is very difficult to understand and predict, but at the same time, there might be some hope in future understanding more of the genetic underpinnings of host specificity, and the accessory genome should definitely not be forgotten as we're trying to continue to investigate it in the future. All right, thank you so much to my lab and for EFSA for bringing me here. Yeah.
Thank you. Is there any question for Alexandra? No, I've got one. I wondered if you want to also take into account the area of origin of the hosts as a co-occurrence of your ancestral Xylella strain and the occurrence in the same area of your potential hosts, as some hosts were introduced in Americas, for example.
Yeah, that's something that I find really interesting. Overall, in Xylella plant relationships, we see very, very few examples where there's a native host as well as a native Xylella strain. Pretty much all of the prominent agriculture, we don't consider it introduced or invasive species, but most of it is plants that have been brought to particular regions because they work well. So almost every single relationship, out of the 349 genomes we had, I'm pretty sure that 348 have either a strain that has been brought to a new region or a host that has been brought to a new region, or at least intensified due to agriculture. So it's really definitely very important for understanding host range.
Is there another question? Over there, Leonardo.
Hey, Alexander, very good idea. My question is, you know, when you show the ancestral state reconstruction that is done in birds and other things, you know, those people in zoology and entomology, they have very good collections that go back many, many years. Now, in terms of Xylella isolates, you know, the one that we use, the oldest was from 1999. You know, do you know if the stuff that Mike Davis got in the 70s or people in the 80s, do we have access to those collections or even plant samples that they were infected before? Do you know if those materials are available? Because that will help you a lot in your project.
Yeah, none of them have been really sequenced that we can use in this whole genome work that I know of. But in general, I think it would be really, really important to add more ancestral strains. Unfortunately, there are a lot of strains that have just been passed from lab to lab over many years and don't have a great history attached to them. But whenever it's possible, I think it would really benefit both the phylogenies that we're creating to have those ancestral points, but also any work we want to do using the phylogenies. So if you know of any particular strains to add to our data set, let me know. All right.
Is there another question? No? Yeah.
I'm thinking about if the host specifically could be associated with the sap composition.
If the host could be associated with the sap composition. Yeah, so I think there are lots of elements that probably determine the overall capacity between any relationship of host and pathogen. I think that if there was only one factor, whether it was xylem size or pili or something like that, if there was only one element that was delineating this relationship, we would have already figured it out. So I think part of the reason that it's so complicated is that there are probably a lot of factors both within the bacteria and also within the plant, whether it's immune response or what have you, that are determining these really complex relationships that don't tend to follow like a normal host pathogen relationship would, where we can see these co-speciations and shared histories. I think there are really quite a few elements that are coming into play, both genetic and environmental.
Another question? We still have time? No? Thanks again.
Thank you.
So now I invite the last speaker of the session, Eduardo Moralero, from the Empresa de Transformación Agraria from Mallorca, Balearic Island, to present his work on the rethinking of the Xylella scenario in the Balearic Island, what epidemiological, phylogenetic, and dendrochronological data tell us. Thanks.
Thank you, Maria Nix. Okay, my presentation is about the origin, chronology, and pathways of the introduction of the strain we have in the Balearic Islands of Xylella fastidiosa. So, in this work has corroborated other researchers from the University of Balearic Island, the Plant Health Service, and from the Spanish Research Council in Córdoba. So straight to the batch. Since 2016, that was when we first detected the Xylella in the Balearic Islands. We have analyzed about 7,000 samples. We have detected 3 subspecies, and these 3 subspecies comprise infecting 21 hosts, and we have found also 5 sequence types. Okay. So in biogeographic, epidemiological, and evolutionary terms, we can distinguish between the two introductions. One was concerned to the Ibiza island that is related to the pauca subspecies, and the other one is in the Mallorca and Menorca that both are related. We'll explain it later. So very briefly in Ibiza, what we found is we have the subspecies pauca, was the second type, 80 is the new one. It's widespread in the island, and it causes especially a disease that's known affecting the olive trees. But something that is much less known is that it causes in the almond trees the leaf spore disease, and it's widespread as well, this one. So I want to focus on the situation in Mallorca and Menorca, and I will tell you something about the distribution of the two species in the island of Mallorca and Menorca. The subspecies fastidiosa, the sequence type number 1, it's in Mallorca and it's only known in this island and is not known in other parts of Europe. It causes in Mallorca the pure disease. If you want to know more about the pure disease in Mallorca, there have been published a recent publication, and you can see it there. The other important disease it causes is the almond leaf spore disease, the same sequence type. The other subspecies, multiplex, is a new sequence type. Sequence type number 81 is very close to the almond clonal lineage that are related to the almond leaf spore disease in California. It causes also on wild olive trees. The mild disease is not killing the trees, but it affects it hardly. So in Menorca, they have both disease, the almond leaf spore disease and the wild olive disease. And we think this sequence type was, the strain was brought from Mallorca to Menorca, and I will explain it later. So the important thing, what I said at the beginning of the first slide, is that we want to know the origin, the chronology, and the pathway of introduction. So I'm going to give you the answer because I want you to focus on the explanation and the evidence I will provide about this. So our answer is that probably Xylella fastidiosa, both the strains, the sequence type 81 and the sequence type number 1, were introduced from California and were brought in around 1993 with plant material to Mallorca. I explained the evidence, and I will brought this evidence from different fields. The first one from the epidemiology. We will see about the etiology of the disease, how is the incidence, how it's transmitted, and what is the pathology of the almond leaf spore disease. The other thing is a new approach that has not done before is that we are using a combination of dendrochronology with qPCR to have an idea of the time scale of the almond leaf spore disease epidemic. And a more classic one is the phylogeny, using based on core genomes, and we use a maximum likelihood and Bayesian trees. Sorry. So we want to go to the present, and we want to know what happened in the past. So now, most of the people here don't know that in Spain, in Mallorca, there was an almond disease, a very important one, that was causing a lot of almond death and infections. And this, we think it began around 2003 in a focus in Son Carrió. If you see, this was attributed, this disease, to fungi. If you see the list of fungi, something very important is that in 2003, when began the problem of the olive quick syndrome in Apulia, they had similar fungi assortments that we found in Mallorca. So if we want to know the origin of the alma leaf spore disease, we have to disentangle the link between the alma leaf spore disease and the fungal trunk pathogens. So this is what we did. And around in winter 2017, we didn't know if there were alma leaf spore reality, but we predicted that we will find it in the summer. So this is what happened. This is an image in the summer 2017, and we predicted that when the worst temperatures triggering the disease, it will appear, and that's what happened. Appeared for all across the island, and this what says that in reality the alma leaf decline is associated with the Xylella fastidiosa. So important thing is that we have the fungal trunk pathogens and the alma leaf spore disease. We need to know which is the relationship between this. Is the fungal trunk pathogens are related to the disease? Is first the Xylella infection and then comes the fungal trunk disease? Well, I will bring you some evidence about this, but we found that what we attributed to the fungal pathogens were indeed, we'll say, an advanced stage of the alma leaf spore disease, actually. So with this data, we went to the field and counted the number of trees infected in orchards around all Mallorca in 2017. By the other way, we observed images from Google Street Map View, and we could follow this sequence of images that comes from alma leaf spore disease to the diebacks in the branches, and then the decrepitude. So this is in the map. You can observe that about 80% of incidents were detected in 2017, and in 2012, the disease incidence was over 50%, and this is an important question. We found that there were several spreading centers around the island. So this is an image from Google Street Map View. This is in Son Carrió, and this is how the field looks after the battle. You can see that in 2012, you have an important disease in Son Carrió. Other thing we need to know is understanding what is happening with the alma leaf spore epidemic. So we performed some from transmission experiments. We wanted to know how it was spread and short distance spread, for long distance spread, sporadic long distance spread with grafting, and then we were interested to know if the pathogen could infect have an 80% of incidence. We want to know if actually the strain we had could infect the almonds. We did all the experiments. I don't want to show you this recipe because I'm not interested to show you this directly, but I can say you that the experiments and transmission was very good, worked very well. If you want to know something about this, there's a poster in the poster room. And the crafting was not so good. We had only 2 successful transmissions from 13 experiments we did with plants, and with the inoculation, we have some success with the strain ST1 of fastidiosa, but we had some problem with the strain ST81 from multiplex. But anyway, we could, for all of them, we could do the Cox postulates for demonstrating the pathogenicity. So the important thing is know what was first, the fungal trunk pathogens or the or the infection of Xylella. We had a lot of information because for calculating disease incidence, we worked to many orchards, have a lot of list of how it looked, so we could associate the almond leaf spore disease with orchard with trees which showed symptoms of the branch of the fungal trunk pathogens. So we could associate this statistically very well in orchards. Also, we had from our observation from the Google Street Map view a lot of sequence of images, so we can rank all the sequence and do some statistics on this. So we did some ANOVA, Freeman ANOVA by ranks, and we could see that was statistically was significant. The other thing is that we could associate the almond leaf core symptoms preceded branch diebacks in 96 of the times. So I show you this graph because we collect a lot of information from the lab, and you can see here that for the almond leaf spore disease from almond samples, you can see the detection frequency in this graph. In this graph, you can see that there's a seasonal trend. Most of the detections were in the summer, and in the next year, in 2018, you had the same pattern, but was influenced by the weather, but the climate was quite different in 2018. So we wanted to bring this idea to have a model how it moves Xylella in the tree. We want to know which is the radial movement of Xylella in the wood. Something important to know is that the water flow and water nutrients in a tree, most of the water flows in the outer rings of the subwood. So this has an influence, we think, has an influence in the movement of Xylella in the wood. There's a gradient from the inner to the outer. So if we look in detail what we think happens, we can show that in every spring when it's formed the new Xylella vessels, bacteria tries to colonize this new tissue. So we think the movement is only this way. It's restricted from the inner to the outside and not going back. So we can use this property to know a little bit more about the infection. This is a good marker to know when the infection occurred. So as we expected, we plotted. We took from the growing rings. We took samples and measured the quantity of bacteria that were in the wood. So we saw that the woods, the growing rings, younger growing rings, they had more bacterial load measured in CQ than in the outer rings. So knowing this, we went we use more trees to analyze this, and here I show you this graph how we've done it. I don't have time. I don't want to explain it too much, but the thing is that the important thing is that you have to mark the growing rings with a stereo microscope, and this is hard work, and then you have to color the rings so that someone who's working with the drill can drill the right growing ring. So taking all these 34 almond trees that were felled, we measured the number of bacteria using PCR. So we have this this data across the 34 trees. And what is interesting is that when you see this infection proportions, it looks like it fits with a sigmoid curve pattern that looks very much to what we think is a polycyclic disease model. And if we take this data and we put it in log transform the data, what you see is a straight line that can be used to extrapolate what the infection were in the beginning of the disease. So this is a rough estimation. You can see that in the intercept with the years, you can see that it's around 1992. So this is it's a year that we will see that is important because it's agreed with something we will explain later. So taking this data, we used the data from knowing how it began the infection to the death of the tree. We can use this to estimate the survival of these trees. So we calculate this, and the median was 13 years from infection to death. Very important is that we found some samples that we can identify Xylella in growing rings from 1998. This one's from fastidiosa, and the other one was multiplex before 2002. Okay, and then we have a lot of information from isolate we have, and we sequence about 10 isolates. We wanted to know the relationship between the isolate from Mallorca and those what we imagined was the most common ancestor was the Californian isolates. So the thing is that the Mallorcan isolates form a single monophylogenetic clade, and important thing is that all the hosts were interpersoned in this clade. So we think that most of the infection were cross-infections, and this agrees with our hypothesis that it was introduced with almond trees and was dispersed through the host. As a aspect for an introduction pathogen, there was little genetic variation measured in the 14 SNPs we found, and in California we have a little more genetic variation as expected because this was the source of the probable introduction. So to conclude with just a moment. Our hypothesis is that in 1992 it was introduced because there was a visit from a delegation from the almond cooperative from Mallorca, and coincidentally, this were coincides with the years we think this could be the introduction. Other thing is that there was long distance dispersal with grafting, and short distance dispersal with the Philenus. So this is what we think happened at the beginning of the disease. There was a spillover from the almond to other hosts like the pear disease and other hosts. This was a cryptic period between 1992 and 2003. That is the time that normally there's no, the alien pathogens needs to build inoculum. So in 2003 appeared the first case of almond decline in San Carrillo, and in the following years there was an experiment, the exponential spread of the disease, but this was attributed to the fungi. So in 2016 was detected for the first time Xylella. So we were about 25 years without the bacteria was overlooked. That's all. Thank you, and sorry for the time.
Thank you very much. We do not have time right now for the question, but anyway, we are just having a time of questions for everybody. So just stay here, please, and I invite all speakers from the session for this question and answer session, part of the session. So I just have a point to mention before we begin to discuss, is that we have a group photo just before lunch. So when the discussion session is finished, then we have to go downstairs and near the stairs for the group photo. So please just group there for the photo. It will be nice to have everybody on the picture. So thanks a lot for your very interesting and very complimentary talks. So now we can discuss all together. So is there any question? Yes, please, I was sure. So, okay, Yanda first, and then, yes.
Yeah, thank you. So it's a question for Eduardo Moralejo. Looking at your results, I'm wondering, what is the role of grafting, not just on this long distance from California or whatever to the Balearics, but indeed in the Balearic Islands, in the particular situation of Mallorca, what would be the role of grafting? And I have a second question, if possible, that is, I would like a little bit more to know how you estimated the incidence. Thank you.
It works, yeah. Thank you for the question. Okay, the first one that was grafting. We think was a sporadic grafting. This is very usual in Mallorca that people like to graft. And when you want to grow a new field of almond trees, people, the farmers, want to know which variety they want to know, and they try to graft in other hosts. But on the other side, there was a gene bank of almond varieties living in Mallorca. There were 4 places, and we know that probably some of the grafting was used for going in other orchards. So there was sporadically spread with grafting, and we know that in the maps we found a lot of spreading centers, not only one in the main one in Son Carrió. And the other question was how we calculate the incidence. In 2017, we went to the orchards. We counted about 30 trees, and we estimated if it was infected by the symptoms. So we said, okay, 30, this is infected. This one. So we did about 121 orchards, and then we used with the coordinates to make the map.
Thank you. Sorry for stressing on the recovery. So I would like to have a clear definition of recovery. Is it a recovery from symptoms and then the bacteria reappears or so stay there at lower population size? And what happens if you have a recovery from symptoms the next years? So the second year, what happens? So the symptoms reappear?
Okay, so the way that I defined recovery for the purpose of my experiments was if the new growth on the plants was tested negative for the pathogen, there were no symptoms, and for a period of 6 months, there were still no symptoms and still no detection of the pathogen. So I cut it off at 6 months because I was working with potted grapevines, and so, I mean, there's only so long that you can keep them. Out in the field, it's possible that you could get a return of the infection over a long period of time. I don't know I didn't really test for that, so I don't know what the very long-term results would be, because this was based on smaller plants.
Yes, please.
We've looked into recovery as well in the field in Napa, and we've seen a lot of recovery in the field, much more than was expected. And over the course of an experiment we've been doing monitoring for 3 years, everything that recovered after the first winter stayed negative, if that's any help.
Yes, thank you. I have a little question for Eduardo and a comment. Because the fungi reported by Gramaye co-workers are well-known primary pathogens of almond and stone fruits. So today we cannot say that that work escaped the infection of Xylella fastidiosa, because all the wood-degrading fungi take a lot of time to give some symptoms. Neophysicoccum and other species are well-known wood-degrading pathogens of almond in California. So, but I have not seen the results from co-inoculation of fungi and Xylella compared with fungi and Xylella inoculated alone, because based on our experience on olive, we were able to demonstrate that fungi alone give just a little contribution to the symptom severity. Probably because they have another time span to show the symptoms. So what are your thoughts about the co-inoculation of fungi and Xylella?
We would like to do it in the future, but you have to take into account the tree age. It's very important, because it has nothing to do inoculation trees of 3 or 5 years and extrapolate these results to a tree of 25, 30 years. We know that tree age is very important. But what what we think is that all these fungi are known endophytes, and there are endophytes that are, when they are activated by some kind of disturbance, they activate to the pathogenicity stage. So it will be important to know this, which is the synergic between the Xylella and these fungi, but it will be a very difficult experiment to do.
Sure, I mean, the time that the fungi require to give some kind of symptoms on plants are not, the same as for Xylella. This is our opinion for olive trees.
I must say that for all these fungi, there was work from Diego Olmo. He did inoculation, and he demonstrated the Koch's postulate for this fungi. They all were pathogenic, but this does not mean that they are causing the disease we're seeing. This is an important question.
We need the pathogenicity test.
And I think we will never know it if we don't have experiments for 10 or 20 years.
Another question? Yes.
Yeah, thanks. It's true for Eduardo. I really like the idea of the rings. It's pretty awesome. So my question is twofold. First is how did you control for contamination between drills? Because it's a practical question. Because I think a lot of people would want to use the same approach. At least I would. And the second one is I really like the regression. It's just that usually for epidemics or invasions, you do have a lag period. before you actually see the invasion or the population take off, in this case, an epidemic. So there is the assumption that the beginning, the introduction itself happened years before you actually saw this really nice curve. So I want an answer for the first one and a comment on the lag period for the second one. Thank you.
Okay, thank you. Good question. I didn't explain that, and I should do it, because when we try to drill and take the sawdust for doing this DNA extraction, we should be careful with the probability that could be some contamination. So to avoid this, we went from the inner side to the outer side, because we thought that the concentration of DNA will be lower, and effectively was this way in the inner side. So every time we drill, we took the sawdust, we clean it very clean with some alcohol. This does not mean that you can have some contamination, but the important thing is the pattern, how you can find that there's a pattern, a gradient from the outer side of the rings to the inner side of the rings. And the other question, yeah, we can only roughly estimate with the data, because we only have 34 trees for estimation this linear regression, and extrapolate to the year. But it agrees very well with what we have with the evidence we have from other circumstantial evidence from a visit from Mallorca. But if you think it very well, how two strains from only known in California, how arrive at the same time to Mallorca. So the idea that was brought from plant material for grafting is the most plausible one. It could be other ways, but we think it's the most plausible one.
Another question? I've got one. You mentioned that you made some transmission experiment. Can you elaborate a little bit on that, please?
Yes, we did some inoculation experiment with the almonds. In Mallorca, we have more than 67 cultivars of almond. So if you want to explain how the Xylella could cause an 80% of disease incidence, you need to demonstrate that it can infect a lot of varieties. We did it with the strain ST1 of fastidiosa, and it works partly well, because not all, we expected all the trees, all the varieties be infected, but only half of them were infected, and we could isolate it and detect it by PCR. But with the subspecies multiplex, we had a lot of problems. Only 2 plants, about 160 we tried to infect, they get really infected.
It was inoculation with the pinprick method or by insects?
It was by the print, yeah.
Okay, thanks. Other questions? Yeah.
I have a general question. I'm wondering something, but I'm not sure if I'm correct. I mean, is it possible that 2 different strains or subspecies of Xylella occur in a vector or in a host?
Yeah, so we have seen 2 different subspecies in a plant, in almond, and we've also seen evidence of 2 different subspecies in insects, which is based on PCR.
We saw this in Mallorca.
Other question? I want to ask a question to... Is there a question? No? Oh, sorry, excuse me. Yep, go.
I have a question for Eduardo. I was interested by the model you talked about inside the xylem vessels. I was just wondering what exactly you took into account for the model. Were you just looking at the free bacteria, or did you also include like biofilm formation?
Yes, this is an assumption. We haven't demonstrated this occurring, but if you see the presentation of Steve Lindo yesterday and how the movement of the bacteria is, you can do it if you try to see if there's live bacteria in the growing rings form in the spring, and see if the in the growing rings, the former growing rings, the bacteria is dead. This is what we want to do to demonstrate this.
Another question? No, I've got one for Miguel. You mentioned in your biofilm tube assay that your Spanish strains were more able to form planktonic cells than attached cells because you had trouble in your solution. So I wondered if you had a look at the DSF production by these strains compared to the other strain you had. Yes, I wondered if you measure the production of the DSF. So no, and did you look at the genome sequences if all the genes coding for the DSF, so the RPF gene cluster, is correct? Is not yet. You will. Okay. Okay. Thanks. I've got a question for all of you. I wondered if you all have already transferred your results for the management of the disease you are working on, and that's a question that can apply to all the situations you manage. So I wondered how you managed to make this transfer, how you worked with the people that can apply the results of your research.
That's my what I'm doing there. When you have strong evidence that the transmissions of the bacterium by plants to planting, and we know by the genetic data, but have strong evidence that the population that you have in the place are the same that in another place, and you know that the main source of one of the place is one not a grover, but it's one place that spread the nursery plants for the all over the place. We immediately talk with the guy for take care about this one. Was not a comfortable situation for me in the first time, but then after they can understand what you are talking about to him, and they stop to sell the nursery plants, and they are redoing all the system of the plant production system, and you are helping him to check the bacteria for the mother plants, and use and start to do a new system to production plants, okay, free of the bacteria.
Is there anyone who wants to elaborate on that, on the transfer of the results directly to the management of the disease? Yeah.
I think one of the easiest ways that the things that I've looked at is applicable is just looking at different cultivars and having a better understanding of what we mean by tolerance or resistance and how it can be influenced by the climate where you're going to be planting these different cultivars.
Yeah, I really, with this particular project, haven't yet been able to produce anything that I feel like would be relevant. I think it would just be so vague, it would be like saying nothing. But I really hope that with this kind of information about host associations, hopefully in the next few years with there'll be enough progression that we'll have better ideas in terms of predicting new outbreaks and susceptibility. So it's always a goal.
Direct implications. There's one very important, that the disease was overlooked for 25 years. So if you think a little about this, the plant health implication for Europe, we should revisitate what we're doing with the eradication contingency measures in some areas.
Is there another question? We are just on time. Yes, so we have to go for the group photo. So thanks again to all the speakers of this session.